Hello, and a very warm welcome to today's Cell and Gene Therapy Insights webinar, titled Accelerating AAV Process Development with NCSU BTEC. This is the final session in our virtual event, Biopharmaceutical Analytics Unveiled. I'm Charlotte Barker, an editor at BioInsights, and joining me today are Blaj Bakalar and Shriarjun Shastry, who will discuss rapid, comprehensive analytical solutions for optimizing AAV process development, including titer analysis and determining empty-full particle ratios. After the presentation, we'll have a live Q&A session, so feel free to pose your questions to our speakers using the Ask a Question box to the left of your screen, and we'll try to get to as many as we can during the session. Now, I'd just like to briefly introduce our presenters. Blaj Bakalar has a PhD in nucleic acid biophysics, and he's now the PATfix product manager at Sartorius BIA Separations. Shriarjun Shastry, a scientist at BTEC, specializes in downstream processes for AAV and mRNA development, with prior experience at Regeneron, and is currently pursuing a PhD in chemical engineering at NC State University. So now, without further ado, I'm going to hand over to Blaj to kick us off for today.
Thank you, Charlotte, and welcome back, everybody. We're going to switch gears a little bit from all nucleic acids to AAV-based therapeutics, another very, very popular based therapeutic modality, especially for gene therapies, and it's actually the most widely used viral vector currently if you examine the clinical pipelines. It can deliver genetic materials such as single-stranded DNA to a broad range of cells and achieve long-term and stable expression. But it's also interesting because it doesn't replicate by itself, and that's why it's an associated virus, and it comes in many different serotypes that have different surface characteristics and different target organs, respectively. It's not triggering to the immune system as compared to other viral vectors, and it's in a wide range of clinical application. Currently, there's quite a few therapies on the market already available, and probably more are incoming in the next year or so. Now, if we look at the challenges of AAV production and purification, it has quite a long, I would say, a production and purification process where you need to start with the for usually HEC cells, you expand them in a C train, you go to production scale, you transfect them. Usually, that's the most common approach now. You transfect them with pDNA plasmid, which then makes the cells produce the AAV, and after a certain amount of time, you can then lyse and harvest the cells and then move forward towards purification. And today's talk is going to be focusing mostly on the purification and analytics that can support this purification or post-lysis or post-harvest process development. So if we look at it a little bit more closely, after the cells are lysed, the samples are usually treated with TFF and sometimes also nuclease to remove a lot of the chromatin, after which they're loading on a capture column, in this case showing us our Simultus SO3 column as a solution. And then a second chromatography step is usually required where, especially on larger scales, where product-related impurities are removed, in this case specifically, usually empty capsids that are produced as well as partials if possible. And finally, the full capsids are formulated and become a drug substance. So analysis of these pre-capture process samples is actually quite challenging and usually requires an extensive sample handling, which of course, every time you handle your sample, it can lead to errors. So the closer you can analyze them to native and get results, the easier it is, the better it is. And optimization of individual steps is difficult without this proper analytical insight, especially pre-capture step. While analytical chromatography allows both early process monitoring and high-resolution separation of multiple AAV species post-capture step. So analytical chromatography, a big advantage here with analytical chromatography is that it's a method established, well over 50 years, which has allowed the field to develop multiple detectors also, not just methods, to probe your sample in different sorts of ways. Here, I'm showing a comparison of fluorescence on the left, mouse in the middle, and UV absorbance plots on the right of different mixtures of empty and full AAV capsids. And we can observe that as we mix them in different ratios, going from 0% empty to 100% empty, we can see the peaks shifting from left to right, from right to left. Looking at these detectors, the reason they're so useful is because they allow you to see multiple things happening in your sample. For instance, the UV detector sees all nucleic acids and protein species, as well as, of course, the AAV capsid, which is very good for early stage samples when you want to visualize impurities. But in those samples, you wouldn't be able to see usually the AAV vector itself, because it would be masked by other impurities. This is where Miles detector is very good and very selective, because even in early stage samples, so early process samples, and which are complex, you can clearly see the viral particles, because they're usually the only thing that is large enough that can scatter light and produce a signal. While the fluorescence detector here shown on the left again is great, because it gives the best indication of all AAV species, because we can select it specifically to measure protein fluorescence. And of course, protein fluorescence means that we're measuring AAV capsid, irrespective of the fact of if they're full, empty, or partial. To give you a closer look at this, I've shown this chromatogram here, where you can see going from the harvest sample shown here in red to the full enriched fraction shown here in yellow. You can see that we're reducing a lot of protein impurities, because we're looking at the fluorescence detector here. And using a single method, you can track all of these impurities throughout your process, from pre-capture samples all the way to fully enriched full capsids. And this allows you, of course, to have a deeper insight into your process. Now, for less complex samples, so I would say post-polish step samples, so if you're analyzing polish fractions to see how well you are able to separate different species, here in the PATfix AAV platform, we've also included a novel method for AAV8 capsids that can separate not just empty and full capsids, but also partial capsids, and as well as heavy capsids. And this significantly enhances your ability to see these species that regulators want to see removed more and more, especially as AAV therapies move further, while not having to switch to a different analytical system or something like that. So combining these, you can basically speed up your process development with these rapid analytics, because we provide you everything that's required to do this analytics. So you don't need to send off samples to basically get basic question on, for instance, if you're playing around with buffer compositions to try to improve your recoveries of a chrome step, but rather you analyze sample as they come, and you can have the results within the same day as you're doing your process development optimization run, which allows you to then iterate much quicker. So putting all of these together, we've designed the PATfix AAV platform to be basically fast and while maintaining high resolution, and it can be used for complex samples with multiple detectors to always give you a deeper insight into your sample. And the methods that we include with the AAV platform, we of course validate according to FDA and EMA guidelines on analytical chromatography. And with that, I'd like to hand over to my colleague Tomasz, who will show us how PATfix AAV platform can be used more hands-on.
Hello, my name is Tomasz and I work in the field of AAV-based therapies, specifically on the downstream process team. Today I want to share with you how advanced analytical methods are transforming our daily work and helping us achieve remarkable results. Therapeutic applications of AAV rely on the removal of process and product-related impurities, which pose serious safety threats and burden the economics of manufacturing. The most critical process-related impurities include host cell impurities such as host cell DNA, host cell proteins, and chromatin complexes, as well as plasmids and transfection reagents. Critical product-related impurities include non-potent AAV capsids such as empty AAV, partially filled AAV, and AAV capsids containing host cell DNA inserts, as well as capsid-to-capsid complexes or aggregates, and capsid DNA complexes. In this product walkthrough, we will explore how PATfix, with its multi-detector setup, can monitor the removal of these impurities throughout the entire AAV downstream process. We know that the fastest way to develop and optimize a process is by utilizing extensive analytical insights leading to effective and safe therapeutic agents. PATfix allows me to gain important insights while optimizing process steps, helping me to complete crucial client projects to the desired specifications and timelines. In-process analytical methods differ from QC analytical methods as they need to focus more on speed and throughput, generating the data required to support immediate process decisions that often need to be made on the spot. One of the biggest challenges we faced was the ability to analyze all DSP samples, including crude samples, without having to extensively prepare them for analysis. Having the ability to analyze pre-capture samples has two key advantages that ease subsequent DSP steps. The downstream process is always optimized to a specific starting material with target AAV full capsid titer, volume, and both process and product-related impurity profiles. By analyzing the starting material, such as cell lysate with UV fluorescence and multi-angle light scattering detectors simultaneously, it becomes much easier to adjust the downstream process steps to the quality of the starting material. Likewise, requirement ranges can be put in place to ensure only material with sufficient quality proceeds further downstream. Then there is the optimization of pre-capture process steps like filtration and TFF. A lot of losses and gains can happen during these two important early DSP steps. Using PATfix and the triple detectors, I can easily compare recoveries and impurity removals between different consumables and process parameters to find the optimal strategy. The light scattering detector is particularly important for crude samples as it allows for specific tracking of particles like AAV capsids. At the same time, UV and fluorescence detectors provide valuable information on the removal of all other protein and nucleic acid impurities. tryptophan for proteins and picogreen for DNA detection at the same time allows me to quickly see any residual impurities that might be present. This is enabled by the fluorescence detector that can monitor two simultaneous channels. UV 260 and 280 nanometers wavelengths detect all protein and nucleic acid components in a sample. This combination of detectors, together with the chromatographic separation, makes it very easy to identify and track all process and product-related impurities. This is especially useful since the full AAV capsids, our product, are always visible even with the impurities present. In post-capture samples using the triple detector on the PATfix system, we can precisely determine not only full and empty AAV capsids, but also other subpopulations that are becoming increasingly important to remove. Compared to other analytical methods, preparing samples, even crude samples, is very easy. It usually involves only dilution with loading buffer, which saves a significant amount of time and effort. Using the included AAV standard and method, I can always check that the system is performing to expectations. This makes it much easier to trust and act on the results obtained. Additionally, I can prepare a calibration curve using the AAV standard, allowing me to determine capsid titer in post-capture samples. One of the features I appreciate the most is having one software for running all the hardware, including all of the detectors, as well as analyzing all the detector data obtained for the samples. This makes it easy to analyze results, share them with colleagues, and present findings to higher ups during meetings. Other solutions often require multiple software programs to obtain the information I need. The system and software are certified according to FDA and EMA regulations, which is important for a few key reasons. Access to data is easy to restrict only to the people in the project group, and all the data obtained is safely stored with access and tamper protections. This is a necessary feature in our industry. The software also allows me to connect from the office, which is great for checking in on progress remotely and analyzing results without having to be in the lab. This flexibility is invaluable for maintaining productivity and ensuring timely decision making. Since I'm using the same package of system, column, buffers, and method that has been validated by my colleagues, I don't have to worry about my analytical results being skewed. This helps me focus on my important work of optimizing the purification process for AAV-based therapeutics. PATfix enables real-time monitoring of AAV samples, allowing me to track changes in impurity levels and the ratio of empty to full AAV capsids throughout the purification process. This real-time data helps me identify potential issues early so that I can make timely adjustments to the process. The ability to monitor these parameters with minimal sample handling required is not only a quality of life improvement, but also helps prevent errors. In turn, it is much easier to streamline the purification process and reduce the time and cost of producing high-quality AAV therapeutics. Rapid chromatographic analysis of AAV fractions removes a bottleneck in fast process development. Sartorius bioseparations has developed the PATfix system specifically for the analysis of large biomolecules and in-process control of upstream and downstream process steps. With its validated methods together with orthogonal analytical approaches, the PATfix system offers a comprehensive solution for insight into the AAV process. It enables fast and precise process monitoring, which is crucial for the successful optimization of gene therapy vector production processes. To learn more about the PATfix AAV platform and its capabilities, visit our website, try out the software demo, or contact our team for more information.
Thank you, that was great. Now I'm going to pass over to Arjun to start his presentation. Arjun, over to you.
Hello, everybody. Thank you, Charlotte. Before I get into the topic of today's webinar, I just wanted to briefly talk about the center that I work for. We are the Golden Leaf Biomanufacturing Training and Education Center located in Raleigh, North Carolina. We are part of the NC State University. There are a lot of different things that we do here at BTEC with the main goal of education and training. We have a master's program. We also offer minor for undergraduates. And for professional industry professionals, we have multiple short courses, such as in downstream, upstream, analytical, as well as some special topics such as gene therapy and automation and so on. We also offer contract services. We have basically any companies or individuals who have ideas, don't have equipments, we typically execute projects for them. Um, Again, in all the fields such as upstream, downstream, analytical, and so on. We also get quite a bit of funding from different institutions, organizations, as well as NC State to do a lot of bioprocess research. That's how this particular gene therapy project has been progressing. The main focus of our center is on biological products for treatment and prevention of human diseases. These are some of the pictures of our facility and our lab space. But if you were to take any of the short course, you would be doing using these instruments hands-on. And if you want to learn more, please visit our website. Or if you're in this area, contact us and we are happy to show you the facility. Now, getting into the topic of today's webinar, I would like to briefly or initially talk about the research that I've been doing for the past few years, and then move on to tell you guys how in the recent past we have utilized PATfix to streamline our research process. My research was focused on developing serotype agnostic peptide ligands, affinity ligands for universal purification of AAVs from the cell culture fluids of HEC293 as well as the SF9 harvest. I work closely with an initiative at NC State called NC Viral. We work with different viral vectors such as lentivirus, adenovirus, adeno-associated virus, and so on. But the main focus of my study was as adeno-associated virus, which are basically the smallest viral vector, have the ability to encapsulate about 4.7 kilobases of single-stranded DNA. And they have a very nice property of tropism, because of which their preferred mode of delivery is in vivo. And some viral vectors are also delivered ex vivo, wherein the cells of the patients are taken, genes are transferred, and the modified cells are put back into the patients. Looking at the current state of art for AAV production process, there are multiple steps involved in upstream and downstream processes, all the way from the seed vial to bioreactor to different chromatography steps and TFF and bulk fill. The main focus of my work is on purification aspects, and I focus more on chromatography. On the right side, you can see there are a bunch of different resins that are currently in the market. Their reported binding serotypes, their approximate cost per liter, and their binding capacities, as well as some of their lifetimes. There are multiple challenges that the industry faces in designing the purification processes for these AAVs. The two most important ones that we targeted in our studies were the prone to loss of transduction activity upon long-term exposure to low pH. All the affinity resins that are currently in the market typically utilize low pH-based elution, due to which the integrity of the capsid or the structure of the capsid can change and come back to its normal state when neutralized. And all this could be detrimental to the capsid's transduction efficiency. And the second one is the high cost of these affinity resins that are in the market. So in order to do that, we started exploring some of the properties of AAV. One of the very interesting property of AAV, like we spoke about, is tropism. In order to show tropism, they have to go to a specific tissue and bind to it. In order to attach to these tissues, they attach to two different types of receptors. One is called primary and the other one is secondary receptor. And the secondary receptor is the one that typically helps engulf the capsid into the tissues. And if you look on the right side, there are a bunch of receptors that are listed, but if you pay attention, there is a receptor called AAVR, which is the adeno-associated virus receptor, which is one of the receptor that most of the AAVs tend to bind to when they're attaching to the tissues. And this was very interesting for us. So we started doing some studies behind this interaction between AAV and the AAVR. And we did a bunch of in silico modeling studies, specifically molecular docking studies with AAV2 and AAVR. Uh, and then we discovered a bunch of peptide-based ligands that could have the ability to attach to AAVs while doing some purification work. We also explored another interesting molecule called A20, which is an anti-AAV mouse antibody, which has the ability to interact with intact capsids rather than the fragmented capsids. So we utilized these two molecules, two interactions, did a bunch of studies, and then figured out a sequence of multiple sequence of peptides that could have the potential to bind to AAVs, and which could be used for doing purification studies. The other interesting thing was they were all modeled in such a way that they could bind at neutral pH, and also the elution can happen close to neutral pH environment. So these were some of the peptides that we had discovered. I just wanted to show you guys that we also looked into their interaction with different AV serotypes. So all the colored cartoons in this slide are the different peptides that were discovered, and all the gray cartoons are the different VP1s of different AAV serotypes. It was interesting that the dissociation constant of these interactions at pH 6 was about 2 logs higher than that of at pH 7.4, which gave it a very nice little nudge to desorb AAVs from the column. So once we had discovered these peptides, we started synthesizing them on the resin beads, and then packing columns and doing some purification studies. So before we started challenging them with lysates, we wanted to make sure they can actually bind to AAVs. So we just ran some pure AAVs. First, obviously, was AAV2, which is what the discovery was performed on. On the left, you can see a nice little structure of AAV2, followed by a few products that are currently in the clinical trials based on AAV2. There is also an approved product in the market, Luxturna. It's a very popular serotype, a lot of studies going on in this particular area. And if you look at the histogram, that's the purification studies that we did. The first bar indicates the feed sample, and then we are seeing all the step yields from different peptides. I've listed only a couple of peptides that were more competitive to the ones that are to the AAVX and AVB resins that are currently in the market. So the number in red basically indicates the step yield. So you can see we got very nice step yields for different peptides. So once we knew that, yes, these peptides have the ability to interact with, that the peptides have the ability to interact with the AAVs, we wanted to challenge them with cell culture fluids. And then we started doing some purification with the HEK as well as the SF9 cell lysates. On the left side is the data for HEK cell lysate. You can see that again in the red are the step yields, and now in this case the blue numbers are basically the log reduction values for the host cell proteins. And you can see we did a pretty good job even when we are comparing it with the commercially available resins. Similarly, the same was the result for the SF9 cell culture fluids as well. We then wanted to make sure whatever we are doing is actually effective. And then we did a transduction assay with human epithelial cells, HT1080 cells, wherein we took the LUHs from the different peptides and then transduced the cells. And you can see the green bars on the histogram here indicate the 3 different peptides that LUHs that we tested, followed by the blue bar, which is the AAVX solution, which was neutralized after it came out of the column. And then the last one, the red red AAVX LU8 was the sample that was never neutralized. So all we wanted to figure out was, or we basically understood that keeping the capsids happy at neutral pH environment is the way to go and to keep the capsids happy here. We also wanted to look into the quality of the capsids just to make sure they are not fragmented or they are all intact, not forming aggregates and so on. We imaged them under imaged the eluates that came out from the column under the mini transmission electron microscope, and they all looked very well intact. We didn't see any big issues here. We also looked at the particle diameter, and they were all distributed between 25 to 30 nanometers in size, which is what we expect for a typical AAV diameter. Finally, we also looked into the dynamic binding capacities of these peptides. These peptides were attached to the toyo pearl resins, and then when we did the dynamic binding capacity, we challenged them with AAV2 in cell culture lysates, such as HEC293, and we found most of the peptides exceeded 2e to the 14 in the 10% breakthrough dynamic binding capacity per ml of resin. Very comparable to what we would see with the commercially available resins as well. And they also seem to maintain their binding capacity even post 20 cycles of usage. All the data that we saw until now were typically from different assays that were all plate-based assays, wherein you determine the concentration of a sample targeting a specific molecule. But what exactly is present in the whole sample is much more better visualized when you get some visual confirmation. So that's when we started integrating HPLC based methods, and that's when we started looking into the integrating the PATfix platform here. This is an image of the system that we have here at BTEC. The very interesting thing about the system is its ability to have multiple detectors in line. So by just injecting one sample, you can get a lot of information about the sample. The nice thing in our system is we have UV, we have mols, we have fluorescence, we have refractive index detector as well. We can get multiple information through that. And we also have a fraction collector, if you look up there, which can, like if you're running critical process samples, if you want to collect it, we have that option as well. One very cool feature that I like about the system is it also has pH and conductivity sensors in line, which very well helps us when we are running gradients. You just don't rely on percentage A and percentage B to make assumption that the gradient is running fine. You can actually see that gradient forming by the change in conductivity traces and the pH traces. So we utilized the PATfix platform to run some methods that we have been working on. One was the size exclusion chromatography. We utilized an interesting 200 Armstrong pore size column, wherein in a 200 Armstrong column, you will not see the AAV go through the pores. They will just flow through the void space, and you'll get a AAV peak initially, and then all the host cell proteins and impurities just go into the pore size and come out much later. So you'll have a pretty good baseline separation between the AAV peak and then the other impurities. So if you look at the first sample, in this case was the feed lysate sample. You can see a nice AVP coming out initially, and then you have multiple impurities. And then later on, when we ran the elution samples from different peptides, we got very nice single AV peak and tiny blips of impurities, which were still lingering there. But this clearly was the visual confirmation that I was talking about, wherein you can see the purification actually happening, right? So I'm pretty sure that the peptides are actually doing the job they're intended to do. We were using fluorescence detector in this case. I think I have the excitation and emission switched in this case. It was excitation at 280 and emission at 350. Also wanted to show you that it works across the board. We tested different serotypes. I did not present all the serotypes here today, but we worked with different serotypes. There was one of the peptide that acted as a universal binder, and we ran all the AAV serotypes through the columns, and we were able to get similar results with most of the serotypes. So just wanted to show you guys that it does not depend on the serotype. It works across the board. And you can see in the red here is the feed sample versus the blue traces are for the elution samples. You can see clear improvement in the or enrichment in the capsids. We also utilized in the recent times to quantitate the amount of capsids that are present using this method. You can do this by building standard curves with standard AAVs, and it works very well and has been very comparable to what we see with ELISA as well. The next method that we explored was the steric exclusion chromatography with the CMAC hydroxyl-based column. The concept of steric exclusion is very well explained by Pete Gagnon in some of his research articles. But basically, it's similar to a PEG precipitation technique wherein the larger molecules in this case tend to get retained back onto the column, and the small molecules such as the host cell protein and impurities just flow through, and you get two different peaks. Basically, if you look at this chromatogram here, the initial big peak on the first sample that is designated as A, you can see a big peak coming off initially, which is the host cell protein and impurities, and then you get a tiny peak of AAV. But when you run the elution samples, you can see clear enrichment or enhancement of those AAV peaks versus the impurity peaks. So clearly there are two peaks which are telling us if we have impurities or if we have reduced the amount of impurities and how much AAVs are still present in the column. So initially we started off with a 30-minute run, and we have now shortened these methods to about 7 to 11 minutes. So in theory, we can quickly get data off of a sample or information about a sample in about 7 to 11 minutes. Again, I just wanted to show you guys that it works across the board. Different serotypes works. Everything can be tested on this particular method. One more interesting thing about this method is a student worked on this particular method. He was the one who actually designed the steric exclusion method. He injected different amounts of samples through the column and basically lysate samples, and designed a standard curve and got a very nice standard curve. So all I want to say is we can quantify the amount of AAVs in the feed sample, which is one of the most challenging things to do. If we do a plate-based assays, there are matrix effect and so on, but this assay can clearly give a baseline separation between the lysate as well as the AAVs, so you can build these standard curves and quantify the amount of AAVs as well. The next method that we are exploring, or we're still under developing this method, it was the ion exchange chromatography with the CMAC empty full. We are still just utilizing the method that was given to us by Sartorius BS Operations, and we have not completely separated the empty and full. We don't get the baseline separation yet, but the initial method itself is giving us a lot of information. This was for AAV8 sample. That is where the method was initially developed. On the left side, you can see the two chromatograms wherein we show the elution samples that were tested. We get clear separation of empty and full, and we can quantify the amount of fulls very nicely. And the one on the bottom is one of the standards that we tested. And we tested this with multiple orthogonal methods, and all of them seem to correlate well with each other. And we have gotten pretty good comparison, and we feel that this method is very reliable. And also looking into some of the initial presentation by Blaž, we can say that it is a very exciting method because they are developing this method to quantify the amount of percentages of fulls in the feed sample, which is again one of the most challenging things to do here. Finally, I'd just like to acknowledge all those people who have one way or the other helped me in this research work, as well as doing different purification techniques here. And then thank you so much.
Thank you, Arjun. Excellent presentation. So before we start our Q&A, we'd just like to ask the audience a quick question, which should be popping up on your screen about now. So we'll just give you a few moments to answer that before we start on the Q&A. Brilliant. Thank you very much for your input. So now we're going to start our Q&A discussion. Do keep your questions coming in, and we'll get to as many as we can and answer any others by email. Wonderful. Okay, so the first question that I have here, what is the biggest challenge in the AAV production and purification processes at the moment? Blaj, do you want to go first?
Sure. From what I hear from industry practitioners, it's mostly to do with increasing yields, first of all, because titers are still on the low side, I would say. And secondly, in the downstream, either completely avoiding or successfully removing the product-related impurities, which are the empties, the partials, the heavies. That's usually the biggest challenge because they can have off-target effects and also are immunogenic.
Perfect.
And Arjun, what would you say to that one?
Yeah, I totally agree with that. I think the biggest challenge that I think is enriching the percentage of full capsids, especially getting rid of those partially filled, and also the capsids that are filled with wrong genome. It's one of the biggest challenges that everybody faces. These contaminants, they can act as decoys and could potentially or essentially cause negative effects to people.
Yeah, absolutely. Next question I've got here. How has the AAV production and purification process changed in the past 5 years with multiple therapies now available in the market? And what do you predict will happen in the next 5 to 10 years? So if you could gaze into your crystal ball, please. Blaj, do you want to go first again?
Sure. So since everybody's moving to higher and higher titers, in the future, I would say an interesting development is probably going to be stable producing line clones, since this would also imply that the therapies they're supposed to treat are broader in reach, because producing a stable clone, of course, is going to require much more effort than currently with transfection. Arjun, what do you think?
Yeah, interesting. Yeah, but basically looking at how it has changed in the last 5 years or so, I would say there were multiple optimization that have occurred, improving like from adaptation of cell from adherent to suspension, improvements of transfection agents to improve plasmid delivery. There's been a lot of capsid engineering to basically avoid the neutralizing antibodies. So many different chromatographic methods with hydroxide stable resins now being developed as well. And of course, there's always improvement in the analytical assays that have been going on in the past few years. But looking like forward, I think there are so many things that can be done. We are still in a phase where like we are 25 years behind if you look at how antibody was being discovered and how challenging the phase was. I think we are in that kind of phase right now. There'll be like cost-effective production systems that people will work on. And there'll be huge demand for these capsids for sure in the future. We'll see more capsid engineering, and they'll probably be more tailored towards the patients as well. And obviously the ligands that I'm working on, something like that works will also go on those kind of things. And I think like you guys are doing the real-time analysis of AAV capsids, percentages of fulls, empties, and all that. I think that's also something that will be done quite a bit in future.
Yeah, interesting that you bring up maps. Like I know from the field that currently the challenge isn't so much in producing high map titers. It's now
Right.
more of how to capture all of them efficiently.
Correct. Makes sense. Yeah, I think that's what we are moving towards. We want to standardize the process, and hopefully we'll get there at one point.
Yeah, let's hope so. So a question here for Arjun. Based on the analytical assays developed, is there a need to create a standard curve for every serotype?
Yeah, yes, that's a good question. So basically every serotype is different. The amino acid sequence identities are very different. So the response that the detectors will offer will vary quite a bit. So yes, we would definitely need separate standard curves for every serotype. Secondly, I would also recommend creating different standard curves if you change the gene of interest as well.
Perfect. And another question for you here, Arjun. Have you quantified partially filled capsids with any of those methods that you spoke about?
No, at present, none of our methods actually work for the partially filled capsids, but when we saw presentations or that video that we just saw, that is a very exciting thing. And I think if we try to start implementing those methodologies, it'll be great. We'll probably be able to quantify the partial and full AAVs. I know mass photometer is one other system that people also use to quantify partial capsids, but I don't have that system, so I don't want to comment more on that performance.
Brilliant, thank you. I think we'll fit in one more question. What other orthogonal analytical techniques have you used to compare the results obtained with the HPLC method, Arjun?
We have run ELISA PCR. That's one of the most commonly used methods. We have, for some samples, the purified samples, we have also tested them on AUC and also on the Waters SEC moles method. We have utilized all these orthogonal comparisons. There's definitely some variabilities between these assays, but overall information obtained inform us that these developed methods are very reliable for both qualitatively and quantitatively understanding the amount of capsids that are present.
Brilliant. Well, thank you very much, Arjun and Blaj, for answering those questions. We're going to wrap up there for today. Any questions that we didn't get to, we'll reply to those by email. This webinar will be available on demand tomorrow, so look out for an email from us with the link. We'd also like to invite you to take a short survey to provide us with some feedback on today's webinar. The link to that can be found in the Session Resources tab on the left-hand side of the screen, and it'll also be in the on-demand email. So all that's left then is to thank Blaj and Arjun once more for a great presentation, and thank you to the audience for joining us for this session and for the virtual event. We do hope you'll join us again soon.
Thank you.
Thank you.